There's a sound a joint makes when you finally move it after a long time of not moving it. A soft, low sound. Not quite a crack, not quite a click. Something between. You've probably heard it when you shifted in bed after a dream that held you still for too long, or when you stood after a few hours sitting in a way that wasn't quite right. Most people don't think much of it. It's just the body waking up. But it isn't nothing. Welcome, if you're here for the first time, to the Sleepy Biologist, and if you've been here before, I'm glad you came back. This space stays alive because of everyone who's found it worth returning to. Channel members, super thanks. Those who've left a quiet comment in the dark, thank you. If tonight is your first time, maybe something will feel worth staying for. There's something your body has been doing all day that you didn't ask it to do and probably didn't notice. Not a dramatic thing, just a kind of low-level monitoring, cellular, electrical, chemical, of how much you've been using your muscles versus how much you haven't. Your body keeps that count. It has kept it since before you were born, in an evolutionary sense, long before there was any concept of a gym or a hospital bed or a long flight where your legs go strange and heavy. Your muscles are surveilled. That's the blunt way to put it. The subtler way is that muscle tissue is biologically expensive and your body doesn't carry expensive things it doesn't need. So when you stop using a muscle, really stop, for days or weeks, something begins. Not all at once, not dramatically, quietly. And that quietness is part of what makes it so strange when you finally notice it. Most of us have had some version of this experience. You've come out of a period of not moving, a bad illness, a healing injury, a stretch of months where everything went wrong and you stopped going outside. And the body you came back to felt different, slightly less solid, a little slower to respond. You probably blamed yourself, or blamed the illness, or just chalked it up to getting older. But underneath that reasonable explanation, there was something more specific happening. Your body wasn't waiting for you. It was adjusting. There's a protein called MERF1, muscle ring finger protein 1, and it belongs to a family of enzymes that tag other proteins for destruction. When muscle activity drops below a certain threshold, MERF1 gets more active. It's not reacting to pain. It's not reacting to damage. It's reacting to the simple absence of the mechanical signals that muscles produce when they contract. When contraction stops, MERF1 gets louder, in a sense. And the proteins it targets are the ones that make muscle fibers thick and strong. I find it hard not to feel something when I read about this. Not quite sadness, it's more like a recognition. The body isn't being cruel when it does this. There's no cruelty in it. It's following an old logic that says, don't carry what you don't use. Don't keep what costs energy to maintain if that energy is needed elsewhere. The muscle doesn't know you were sick. It doesn't know you were depressed or traveling or stuck. It knows only what the signals tell it. And the signals say stillness, absence, quiet. The ubiquitin proteasome pathway is the machinery behind most of this. Ubiquitin, a small protein, gets attached to other proteins as a signal, a kind of mark. Marked proteins are then fed into a structure called the proteasome, which breaks them down into their component amino acids. It's happening in you right now, as it happens in every living human cell, all the time. Protein turnover is constant. What changes under disuse is the rate. The balance tips, and breakdown begins to outpace construction. Outside the window, or wherever you are right now, there might be a hum from something electrical, a refrigerator, an air conditioner, something that runs so constantly you've stopped hearing it. That's roughly what protein turnover feels like from the inside. You never feel it happening. You only notice its consequences. When astronauts return from long missions in microgravity, their legs are sometimes too weak to hold them upright without assistance. They have to be helped from the capsule. They've been healthy, well-fed, in an environment of controlled nutrition and careful monitoring. And still, within weeks, their lower body muscle mass drops measurably. Because in microgravity, the mechanical loading that signals a muscle to maintain itself simply isn't there. The floor doesn't push back. The body responds to what is actually happening, not to what the person intends. The signals, not the intentions. I think that's the part people find hardest to sit with, that your body isn't listening to what you want, it's listening to what you do. And it starts adjusting within days of a change in what you do, not weeks, not after some threshold of neglect is crossed. Days. The biology isn't waiting for you to notice. There's another protein involved here, or rather, a pathway. The mTOR pathway, mechanistic target of rapamycin. When muscles are loaded, when they're used, stressed, asked to do something, mTOR signaling goes up and muscle protein synthesis follows. When loading disappears, mTOR goes quiet and synthesis drops. It's not that the body forgets how to build muscle. The blueprint doesn't disappear. The machinery is still there. It just stops running at the same rate because the signal that says keep building has gone silent. Somewhere in a hospital right now, someone is lying in a bed they cannot leave. They are receiving good care, food, fluids, medications. Their heart is being monitored, their blood is being checked, and their muscles are quietly, without drama, beginning to thin. Not because the doctors aren't paying attention, because muscle, at the cellular level, isn't asking whether the situation is temporary. It's responding to now. There's something we still don't entirely understand about the threshold, the exact point at which the body decides inactivity has become threat enough to warrant dismantling. It varies by age, it varies by prior fitness, it varies by things we haven't fully mapped. What we know is that the process is faster than most people expect and slower to reverse than it was to begin. And somewhere in the biology of animals that spend months completely still, genuinely still, not just sedentary, there is something that behaves differently, something that holds. You'll meet it soon. You've never felt atrophy happening. That's the first thing worth saying, not because it doesn't happen, it does, and it's happening right now in some small measure, in the fibers you haven't used today, in the motor units that weren't recruited, in the proteins whose tags are already being read by molecular machinery that doesn't sleep and doesn't wait. But you don't feel it because the process is not designed to be felt, it's designed to be efficient. Efficient is the right word, actually. Not cruel, not indifferent. Efficient in the way an old accounting system is efficient. Thorough, unsentimental, operating on rules that were written long before you existed and will continue long after. The cell doesn't know your name. There's a specific feeling. And I think most people have had some version of it that comes a few weeks after a limb has been in a cast. The cast comes off, the skin is pale and slightly strange looking, and when you pick up your own arm or leg, there's a lightness that is wrong, not a pleasant lightness. Something closer to hollowness, though the limb is still there, still intact. You expected to find yourself inside it. Instead, there's slightly less of you than before. The muscle beneath the skin has been partially unmade, and the body you reach for in that moment is a body that has already quietly revised its estimate of what you need. That feeling is proteolysis, or rather, that feeling is the consequence of proteolysis. The enzymatic breakdown of protein, specifically the contractile proteins inside muscle fibers, that your body has been conducting in your absence for the entire duration of your immobility. Contractile proteins are what make a muscle a muscle. Actin, myosin, the long filaments that slide past each other when a nerve fires, shortening the fiber, producing force. Without them, a muscle cell is just a cell, alive, metabolically active, but structurally emptied of its purpose. Atrophy doesn't kill the cell, it just removes what the cell was for. The ubiquitin proteasome system is the machinery of this removal. Ubiquitin is a small protein, only 76 amino acids long, and it exists in essentially every eukaryotic cell that has ever been studied. In yeast, in plants, in the cells lining your stomach, and the neurons running up your spine. Its job is attachment. It binds to target proteins as a signal, a kind of molecular flag, and once flagged, those proteins are fed into the proteasome, a large barrel-shaped complex that works like a very slow shredder. The protein enters one end and exits the other as fragments, broken down into amino acids that can be recycled or excreted. What changes under disuse is not whether this system runs. It runs always. What changes is who gets flagged. In an active muscle, the balance between synthesis and breakdown holds something close to steady. Proteins are built, proteins are broken down. The net result, in a healthy and exercised body, is maintenance or growth. But when mechanical loading disappears, when the signals that a contracting muscle generates, the electrical impulses, the stretch, the metabolic byproducts, when those signals go quiet, the balance tilts. EMRF1 and MAFBX, two ubiquitin ligases that are somewhat specific to muscle tissue, become more active. More flags go out. More contractile proteins get marked. The shredder runs faster than the factory, and the fiber narrows. Outside right now, there might be rain or traffic, or just a particular silence of wherever you are. And beneath your skin, in muscle fibers you haven't thought about since you last needed them, this arithmetic is being done. Myosin heavy chain is one of the first proteins to go. This matters because myosin is the motor. It's the protein that actually generates force that reaches out and pulls along the actin filament during contraction, consuming ATP in the process. Losing myosin heavy chain is not losing decoration. It's losing the engine, and it goes within days of disuse beginning. Not weeks. The studies on bedrest, on limb immobilization, on spaceflight, they all show the same thing. The muscle doesn't wait to see if the stillness is temporary. It starts adjusting within about 48 hours. There's something about that number, 48 hours, that I've sat with longer than most facts I've encountered in biology. It feels faster than it should, faster than seems fair maybe, though fairness is not a concept the proteasome entertains. The mTOR pathway runs the other side of this. mTOR, mechanistic target of rapamycin, is a kinase, a protein that adds phosphate groups to other proteins, modulating their activity. When muscles are loaded, when they are contracting and being stressed and doing what they were built to do, mTOR signaling is active. It promotes the translation of messenger RNA into new muscle proteins. It pushes the factory to run. When loading disappears, mTOR signaling drops, and with it the synthetic rate. The cell isn't broken. The genome still contains every instruction needed to build myosin, to build actin, to build the scaffolding and the regulatory proteins and the metabolic enzymes. The instructions exist. The cell simply stops reading them at the same pace because the signal that authorized the reading has gone quiet. This is not the same as forgetting. It's something stranger. A library where all the books are still there, on the shelves, intact. But the lights have been dimmed, and nobody is coming in, and after some time the staff begins quietly moving the volumes to storage, not destroying them, just recalibrating around the apparent reality that this particular collection is not currently in demand. I said I'd drop that metaphor. I will. The library doesn't quite hold anyway, because what's actually happening is more physical than that. The proteasome is real and mechanical, and it processes proteins in a way that takes time and energy and leaves specific fragments. The process is not abstract. It is a chain of molecular events, each one following the last, that results in a muscle fiber that has a smaller cross-sectional area than it did before, generates less force, and holds less water, which is part of why the post-cast limb looks both thinner and stranger, a slightly desiccated version of the original. The sensation of picking up a limb and finding less of yourself than you left, doesn't have a clean cultural name. We say muscle loss like it's neutral, like it's a statistic, but the experience of it has a texture that the phrase doesn't hold. There's something unsettling about discovering that absence inside your own body, even when you know exactly why it happened, even when you're holding the cast that caused it. In elderly patients who go through surgery and spend several weeks recovering in bed, the muscle loss from that period alone can take months to recover, if it's recovered fully at all. The biology is not symmetric. Building requires more signal, more energy, more time than unmaking. The arithmetic runs in one direction faster than the other, and this asymmetry is not an accident or a design flaw. It is ancient. It predates the medical system that now works against it. A body in genuine stillness is, from an evolutionary standpoint, probably a sick body, or a starving one, or a dead one. The instructions written into your cells for responding to prolonged disuse were written in an environment where prolonged disuse was an emergency, not a hospital recovery protocol, or a long-haul flight, or a winter of inertia. The cells don't read the context. They read the signal, or the absence of it. This is the arithmetic, not a punishment, not a failure of will. An ancient sum done in the dark of the body that keeps coming up with the same answer, less. And somewhere, in a body that spends months motionless inside a den in winter, that sum doesn't run the same way. The answer comes out different, not less. The same, or close enough to the same that when spring arrives, the animal walks out whole. A brown bear in winter weighs somewhere between 200 and 600 kilograms, depending on the bear, depending on the year, depending on how well the summer went. By the time it enters its den, it has stopped eating. Not tapered off, stopped. The last meal is behind it, sometimes by weeks. And then it curls into whatever hollow or hillside it has chosen, and it becomes still in a way that is genuinely difficult to hold in your mind. Not sleep exactly, or not only sleep. The word hibernation gets used casually, but what the bear enters is something more specific, a state of metabolic depression so thorough that its heart rate drops from somewhere around 40 beats per minute down to 8, sometimes fewer. Its core temperature falls, though not as dramatically as in smaller hibernators. Its breathing slows to something you could count on one hand per minute. Months pass. There is no eating. There is no drinking. There is no urination, no defecation. The kidneys continue filtering blood, but the waste products are processed differently, recycled rather than expelled. The bear's body is doing something with its own waste that a human body cannot do, or won't, or has forgotten how. And here is the part that should stop you. After 5, 6, sometimes 7 months of this, motionless, fasting, at reduced temperature, sealed into a space the size of a large dog's kennel, the bear emerges, and its muscles are largely intact, not perfectly intact. There is some loss, in some bears, under some conditions, but the degree of preservation is so far outside what human physiology would predict that researchers who study it for years still describe the data with something that sounds a lot like disbelief. A human being immobilized for the same duration would be a different person coming out, not in a metaphorical sense, physiologically, measurably different. Weaker, thinner in the limbs, requiring weeks or months of rehabilitation just to walk without assistance. The ubiquitin proteasome system we talked about, the machinery that flags muscle proteins for destruction when loading disappears, would have been running hard the entire time. Myosin gone, actin reduced, the fibers narrowed, the bear walks out. I keep returning to that image, the bear, walking, not rehabilitating, not limping into spring on atrophied legs, walking. And soon after, running, foraging, doing whatever the season requires. The muscle is there, the strength is there. Something in the biology of that animal held what yours cannot hold under the same conditions, what it held and how, and what that might mean for the rest of us. That's not a question this chapter is going to answer yet. Some things deserve to sit for a moment before they're explained. Consider the numbers again, briefly. 7 months, heart rate of 8 beats per minute, no food, no water, muscle, preserved. There is no human equivalent, not even close. The longest Bedrest studies run to about 3 months, and they produce measurable, clinically significant muscle loss. The longest documented human fasts produce catastrophic loss of lean mass within weeks. The conditions inside that den, no mechanical loading, no protein intake, reduced temperature, complete inactivity, are, by every rule of human muscle biology, a perfect prescription for atrophy. And yet, I don't have a better phrase for it than that. And yet, the biology breaks the rule it's supposed to follow, and the animal comes out whole, and the mechanism behind this is real and studied and to some degree understood. But right now, in this chapter, I find I want to let the strangeness be strange for just a moment longer. You've spent 2 chapters learning how carefully, how swiftly, how efficiently your body dismantles what it doesn't use. The arithmetic we talked about, the ancient sum that keeps coming up with less, the bear does not get that answer, and we don't entirely know why. Most of what your body does with protein ends in nitrogen, not immediately. The path is longer than that and involves more steps than this chapter will trace fully. But eventually, when a protein is broken down, when the peptide bonds between its amino acids are cleaved and the individual units are liberated, the nitrogen that was part of those amino acids needs to go somewhere. Your body cannot store free nitrogen. It has no organ for that, no reservoir, no patient-holding chamber, so it converts it. The nitrogen gets folded into a molecule called urea. 2 amino groups, 1 carbon, 1 oxygen, arranged into a small, stable, water-soluble compound. And the urea passes into the blood, reaches the kidneys, and leaves the body in urine. This is the urea cycle, and it runs constantly, in you and in every other mammal, every hour of every day. It is one of the most ancient metabolic pathways in vertebrate biology, older than sleep, older than bone. Running before there were mammals to run it, in some earlier form, in creatures whose names we don't have. The bear, in its den, is making urea too. Protein turnover doesn't stop entirely during hibernation. It slows, shifts, rebalances, but the chemistry of amino acid catabolism continues at a reduced rate, and urea is still being produced. What changes is what happens to it next. The kidneys of a hibernating bear are still filtering blood, but urine production drops to almost nothing, essentially zero in many species for the duration of torpor. The urea that the kidneys would normally clear from the bloodstream, that steady waste product being carried to the exit, has nowhere to go. It accumulates. Blood urea nitrogen rises to levels that, in a human patient, would indicate kidney failure and require immediate medical intervention. But the bear is not failing. Something downstream of that accumulation is using it. Here is where the gut becomes part of this story, which is not where you'd expect the story to go. The gut microbiome of hibernating bears, the trillions of bacteria and archaea and fungi that live in the digestive tract, undergoes a significant compositional shift during torpor. The bacterial populations that dominate during active seasons are partly displaced by populations that are, functionally, more useful in the context of a body that is fasting and accumulating nitrogen-containing waste. Some of these bacteria produce ureas, an enzyme that cleaves urea back into its components. Carbon dioxide and ammonia. The ammonia is then available as a nitrogen source, and certain bacteria in the gut can incorporate that nitrogen into their own proteins, into bacterial biomass. And when those bacteria die or are processed, the nitrogen they've incorporated becomes available again, absorbed back through the gut wall, re-entering the bear's own circulation. Nitrogen recaptured, not from food, from waste. The molecule that was heading for the exit has been turned around, fed back, run through another cycle. The bear's body, in a state of complete fasting, is extracting nitrogen from its own urea and cycling it back into something usable, ultimately through a pathway that involves gut bacteria as intermediaries back toward amino acid synthesis and from there toward muscle protein maintenance. This is the part I find hardest to just move past. Not because it's dramatic, it isn't, in the way that biology described slowly often isn't, but because of what it implies. The bear is not consuming food. It is not consuming muscle at the rate its own inactivity would suggest. It is, in some functional sense, feeding itself on its own waste, with help from organisms it didn't choose to host, through a process it has no awareness of, in the dark, for months. Without consent, without knowledge, without effort. There's a particular silence in that, not a peaceful one exactly, more like the silence of something very old running in a room you didn't know existed. Nitrogen is the fourth most abundant element in the human body. We carry it in every protein, every nucleic acid, every molecule of ATP. Getting enough of it, from dietary protein, from amino acids absorbed in the gut, is one of the central metabolic tasks that every mammal solves anew each day by eating. The bear, midwinter, somewhere under 2 feet of snow, is solving it differently, laterally, through a closed loop that most mammalian biology doesn't bother to maintain because food has always been available. Outside, wherever you are tonight, if there's a window near you, or if you're just imagining one, winter presses against the glass with nothing to show for it. And in a den somewhere in North America or Scandinavia or Russia, a bear's gut bacteria are running a nitrogen economy that human metabolic medicine has only recently begun to take seriously as a model. Studies in the last 15 years have looked at this recycling loop with increasing specificity. Researchers have identified the bacterial species involved, members of genera like Clostridium and others, measured the urea nitrogen flux, traced the incorporated nitrogen into bear muscle protein using isotope labeling. The data is real. The loop is real. The muscle, preserved through winter partly on recycled waste, is real. What that means for human medicine, for patients in long-term immobility, for muscle-wasting diseases, for the biology of fasting, is still being worked out. We don't have a therapeutic equivalent. We don't yet know how to replicate the bear's nitrogen loop in a human gut, and the microbiome differences involved are substantial. But the logic of it exists. An old logic, pre-mammalian possibly, something that emerged in a body that could not afford to lose what it had built, in a winter that did not care whether it could afford to. The nitrogen circles back, and the fiber holds. There is something happening in your body right now that you almost certainly don't have a word for. Not a sensation, exactly, more like the absence of one, a settling, a dropping of some internal register that you probably noticed without naming sometime in the last hour. The slight heaviness behind the eyes, the way sounds from outside became less urgent. The moment your grip on the day loosened, not because you decided to let go, but because something in your biology had already started the process before you noticed it was happening. That process has a hormonal architecture, and the architecture is more deliberate than most people realize. Sometime in the first hour or two of deep sleep, what sleep researchers call slow wave sleep, the stages where brain activity slows into long synchronized waves, where the body becomes genuinely hard to wake. The pituitary gland releases a pulse of growth hormone, not a trickle, a pulse. Rapid, substantial, measurable. The largest single release of growth hormone in the entire 24-hour cycle happens here, in this darkness, while you are not aware of it. While you are, in most practical senses, gone. Growth hormone. The name is slightly misleading, or at least incomplete. In children, yes, it drives longitudinal bone growth, the literal getting taller that happens mostly at night, which is why that particular piece of parenting lore turns out to be genuinely true. But in adults, in the sleeping body you inhabit right now, growth hormone is doing something more specific. It is a metabolic signal, a rerouting instruction. It tells fat tissue to release fatty acids for energy. It tells the liver to produce another hormone, IGF-1. And it tells muscle tissue, among other things, to shift towards synthesis. The body, in deep sleep, tips toward building, not dramatically, not in a way you'd notice if you could watch it, but measurably, at the level of protein turnover, at the level of what the proteasome is doing versus what the ribosome is doing, the balance changes. The machinery that makes muscle proteins runs at a slightly higher rate. The machinery that breaks them down runs at a slightly lower one. The net result, night after night after night, is maintenance, repair of the small damage that accumulates during the day, replacement of proteins that have reached the end of their functional lifespan, occasional addition of new contractile capacity if training has provided the stimulus. IGF-1 deserves its own moment here. Insulin-like growth factor 1 is produced primarily by the liver in response to growth hormone signaling, and it circulates through the bloodstream until it reaches a muscle cell, where it binds to a receptor on the cell's surface. The IGF-1 receptor, a tyrosine kinase receptor that spans the cell membrane. When IGF-1 binds, the receptor changes shape. Not metaphorically, physically, the protein folds differently at the moment of binding, and that conformational change initiates a cascade of phosphorylation events inside the cell. One protein activates another, which activates another, and downstream of all that signaling, the mTOR pathway, the same pathway that goes quiet under disuse, wakes up. Protein synthesis increases. The ribosome gets the signal that it's time to work. All of this is happening while you sleep. The room is dark, your breathing has slowed, and inside the cells of the muscle tissue you use most, ribosomes are running. I find it genuinely difficult not to feel something about this. Not sentiment. The biology doesn't need sentiment added to it. It's just that the image of it is so specific and so quiet. All that cellular activity, organized and purposeful, occurring in a body that appears from the outside to be doing nothing. The stillness is the work in some sense. The inactivity is when the construction happens. There is a hormone called cortisol that will eventually end this window. Cortisol is a glucocorticoid. It belongs to a class of steroid hormones produced by the adrenal cortex. And its levels follow a circadian rhythm that reaches its lowest point in the middle of the night and begins rising in the hours before dawn. By the time you wake, cortisol is near its daily peak. It is partly what wakes you, in a biological sense. It raises blood sugar, increases alertness, prepares the body for activity. This is appropriate. This is the design. But cortisol at sufficient levels is catabolic to muscle. It promotes protein breakdown. It opposes the anabolic action of growth hormone and IGF-1. And so there is a window, a genuine, bounded, measurable window, in the middle of the night, in deep sleep, during which the anabolic hormones are high and cortisol is low, and the body is briefly free to build. The width of that window depends on how long you sleep and how deeply you sleep. cut the night short and you cut the window. Wake frequently through the night, and the depth of slow-wave sleep fragments and the growth hormone pulse, which depends on that depth, becomes smaller or mistimed. Sleep in a lit room or on a device or after alcohol, and the architecture of the stages shifts in ways that specifically reduce the proportion of slow-wave sleep. Not ruin it entirely, reduce it, shorten the window, trim the pulse. The cells still hear something, but not as clearly. I'm not making a case for anything here. People have complicated relationships with sleep, and the reasons sleep gets disrupted are rarely casual. They're stress, pain, young children, anxiety, shift work, circumstance. The biology doesn't grade on a curve, but I'm not going to pretend the solution is obvious. What I want to stay with is just the fact of the window, that it's real, that it has receptors and hormones and measurable downstream effects, that the body has arranged its chemistry around this nightly pulse in a way that suggests sleep is not a passive absence of activity, but an active biological state with its own anabolic agenda. Go back further now. The growth hormone receptor, the one on the surface of target cells waiting for its ligand. When growth hormone binds, the receptor dimerizes. Two receptor molecules come together, held by the hormone between them like a hinge, and this complex activates a signaling protein called AK2, which activates transcription factors called STATS, which enter the nucleus and bind to specific DNA sequences and change which genes are being expressed. The downstream effects of growth hormone are ultimately gene expression changes. The hormone doesn't build the protein itself, it changes which proteins the cell bothers to make. Sleep changes what your genome is doing, not permanently, not in a way that alters the sequence, but in the sense that gene expression, which genes are being transcribed, which proteins are being produced, which cellular processes are running, is different in a sleeping body than in a waking one. The epigenetic landscape of a muscle cell at 3 a.m. is measurably different from the same cell at 3 p.m. Different transcription factors are active, different promoter regions are open. The cell is reading different passages of the same book. Your body, right now, if you're deep enough into this night, is reading the passages about repair. There's something in the biology of deep sleep that rhymes with what the bear is doing, without being the same thing. The bear's preservation of muscle over months is achieved through mechanisms we've only started tracing. But the principle underneath both, that the body uses its quietest hours to run its most constructive chemistry, is not unique to hibernators. It's in you. It's running tonight. The pulse has already happened, or it will happen soon. You won't feel it. You never do. But somewhere downstream of the pituitary, in the dark of a sleeping body that looks from the outside like it's simply resting, a receptor is changing shape. A cascade is beginning. A ribosome is reading. The signals your cells hear at night are not loud. They were never meant to be heard at all. They are old and chemical, and they run in the background of every night you've ever slept, doing something your waking mind has never once had to ask them to do. The window is open. Every cell in your body is wrapped in a membrane made mostly of fat, not a simple fat, a structured one, phospholipids arranged in a double layer, heads facing outward towards water, tails tucked inward away from it, held together by no bonds at all, just chemistry and preference. The membrane isn't rigid, it moves. Individual lipid molecules drift laterally through the bilayer, shifting position constantly, the whole structure remaining fluid enough to allow proteins embedded within it to function, to open, close, change shape, pass signals through. Fluidity is the property the membrane must maintain to work. Too rigid, and the proteins stop moving. The channels close, The receptors can't bind. The signals don't cross. Cold makes membranes rigid. In a human cell exposed to sustained cold, not frostbite cold, just meaningfully below normal body temperature, the phospholipid tails begin to pack more tightly. The membrane stiffens. The proteins embedded in it lose the lateral freedom they need. Signaling slows. In extreme cases, it stops. Hibernating animals solve this before it becomes a problem. In the weeks before entering torpor, during a preparatory phase called pre-hibernation, the body composition of cell membranes shifts. The proportion of unsaturated fatty acids in the phospholipid tails increases. Polyunsaturated fatty acids, including docosahexaenoic acid, get incorporated in higher concentrations. Unsaturated fats have kinks in their carbon chains, literal bends in the molecular geometry that prevent the tight packing that rigidity requires. More kinks, more space between molecules, more fluidity retained at lower temperatures. The membrane that enters winter is not the membrane that was there in autumn. It's been rebuilt, lipid by lipid, to tolerate what's coming. I find this detail hard to move past quickly, though I'm aware it might seem like a footnote. It isn't, because membrane fluidity at low temperature is not only about keeping cells alive, it's about keeping them signal capable. A membrane that can still flex at 8 degrees can still pass hormonal signals through. The growth hormone receptor can still change shape when its ligand arrives. The IGF-1 cascade can still begin. A membrane that has gone rigid cannot do any of that. Whether repair happens or breakdown begins depends on which signals reach the cell. Which signals reach the cell depends on whether the receptors are functional. Whether the receptors are functional depends, in part, on whether the membrane they're embedded in is still moving. The membrane that doesn't freeze keeps the conversation open. What that conversation says, how the hibernating cell uses those open channels to hold what it has built, that's a question for later chapters. For now, the lipid has been changed. The membrane flexes. The cold arrives and finds a surface already prepared for it, already different from what it was, already carrying the molecular accommodation that will let the rest of the biology continue, something so small. The ground squirrel weighs about 250 grams when it enters torpor. It will spend the next 6 to 8 months in a state where its body temperature drops to within a degree or two of the ambient temperature in its burrow, sometimes close to 0 degrees Celsius. And its metabolic rate falls to somewhere between 1 and 5% of what it runs during summer. Its heart beats a few times per minute. Its breathing is occasional rather than continuous. And inside its muscle cells, proteolysis is still happening. Slower, much slower, but not stopped. This distinction matters more than it might seem. The question isn't whether protein breakdown occurs during hibernation. It does, in squirrels as in bears, in all hibernating mammals that have been studied with sufficient resolution. The question is what kind of breakdown, at what rate, targeting which proteins, and whether the synthesis running alongside it is sufficient to compensate. The question is the tilt. In a normally active cell, protein turnover is constant, synthesis and degradation running in rough parallel, the net result being maintenance or growth, depending on what signals are present. The ubiquitin proteasome system handles the majority of regulated protein degradation, tagging damaged or excess proteins with ubiquitin and feeding them to the proteasome for disassembly. In the hibernating ground squirrel, proteasome activity doesn't shut down, it slows. Measured proteasomal degradation rates in torpid ground. Squirrel muscle tissue are reduced by somewhere between 40 and 60% compared to active animals, a significant reduction but not cessation. The cell is still running the shredder, just at half speed. What changes more dramatically is the synthesis side. Ribosomal activity, the rate at which messenger RNA is being translated into new protein drops substantially during deep torpor, more than proteasome activity drops, which means the balance shifts. Both processes slow, but synthesis slows more, and for a time, the ratio tips slightly toward net breakdown. Slightly, not catastrophically, not even close to the rate a human muscle would lose protein under equivalent conditions of inactivity and temperature reduction. The difference lives partly in AMP-activated protein kinase. AMPK is an enzyme that functions as a cellular energy sensor. It monitors the ratio of AMP to ATP inside the cell, a ratio that reflects how depleted the cell's energy currency is. When energy is low, AMP accumulates relative to ATP. AMPK becomes active, and it begins shutting down energy-expensive processes to conserve what remains. Protein synthesis is one of the most energy-expensive things a cell does. AMPK, when active, suppresses mTOR signaling, the same pathway that drives muscle protein synthesis, and with mTOR suppressed, the ribosome slows. In the hibernating squirrel, AMPK activity is elevated during torpor. The cell's energy is reduced. The sensor detects this, the synthesis rate drops in response. But AMPK does something else that is less obvious and considerably more interesting, at least to me. It also influences autophagy, the cellular process by which damaged organelles and misfolded proteins are sequestered in membrane-bound vesicles called autophagosums and delivered to the lysosome for degradation. Autophagy is a different proteolytic pathway than the ubiquitin proteasome system, slower, more targeted toward bulk cellular housekeeping than individual protein disposal. And during hibernation in ground squirrels, autophagy appears to remain active at a level sufficient to clear damaged cellular material, even while the proteasome slows and synthesis drops. The cell is not suspended, it is maintaining. There's a threshold below which muscle tissue cannot go without beginning to damage itself through mechanisms that are harder to reverse. Inadequate clearance of damaged proteins, accumulation of dysfunctional mitochondria, eventual necrosis of fibers that have been deprived of sufficient turnover for too long. The hibernating squirrel's muscle biology appears calibrated to stay above that threshold without spending the energy that full normal turnover would require. Enough autophagy to clear damage, enough proteasomal activity to process the proteins that need processing, enough synthesis maintained at low rate through the torpor arousal cycles that interrupt deep hibernation every 1 to 2 weeks to prevent the deficit from becoming structural loss. Those arousal cycles matter. Every week or two, the ground squirrel briefly re-warms, not to full waking temperature, but up enough to allow normal cellular activity for several hours. During these arousals, metabolic rate spikes. Protein synthesis increases sharply, driven in part by the restoration of mTOR signaling as energy status improves and AMPK activity falls. The ribosome, briefly, runs at something approaching its active season rate, and then the animal re-enters torpor and the cycle continues. The brief arousals are metabolically expensive. They account for a disproportionate fraction of the total energy expenditure of hibernation, far more than the torpor bouts themselves. The squirrel is paying for them. Something in the biology has determined they are worth paying for. The synthesis window those arousals open is almost certainly part of the reason. There's a kind of patience in this that I find hard to fully articulate. The cell isn't waiting for spring to begin repairing itself. It isn't suspending all maintenance until conditions improve. It's running a reduced version of the same careful accounting that it runs during summer. Slower, more conservative, timed to the intermittent windows when the energy to do it is available. The tilt exists. The deficit is real, and the biology is managing it week by week, arousal by arousal, with a precision that requires no awareness and no effort from the animal sleeping above it. Synthesis slow, degradation slower, autophagy running, the tilt held. Starvation and torpor are not the same thing. This seems obvious once stated, but the distinction is easy to collapse. Both involve prolonged absence of food, both involve caloric deficit, both involve a body running on something other than what it ate for breakfast. The overlap is real, but the underlying physiology diverges in ways that matter considerably for what happens to muscle, and collapsing the two is how the bear's winter gets misunderstood. In a starving human body, the sequence is fairly well established. Glucose runs low within the first day. Glycogen stores, the glucose polymer held in liver and muscle, are depleted within roughly 24 hours of fasting, sometimes faster depending on prior activity and body composition. The liver begins producing glucose from non-carbohydrate sources, glycerol from fat breakdown, and amino acids from protein. That last source is the problem. Amino acids come from somewhere, and in a fasting body, they come partly from muscle. Gluconeogenesis, the synthesis of new glucose from non-sugar precursors, draws on muscle protein as a substrate, and the longer the fast continues, the more muscle is consumed to sustain blood glucose. The body, in genuine starvation, turns to its own structure for fuel. The hibernating animal mostly doesn't. Not because it has found a way around the need for energy, it hasn't. It still requires ATP. Its cells still run ion pumps and maintain membrane potentials and perform the minimum housekeeping chemistry that keeps them alive. The difference is where that energy comes from, and the answer is fat. Almost exclusively, deeply, specifically, fat. In the weeks before entering torpor, hibernating mammals accumulate large fat reserves, not incidentally, but as a precise biological preparation. Body fat percentage in pre-hibernation bears can exceed 40%. The fat is not stored and forgotten. It is stored and then accessed with a specificity that human fat metabolism during ordinary fasting approaches but doesn't fully replicate. During torpor, lipolysis, the breakdown of triglycerides stored in adipose tissue into glycerol and free fatty acids runs at a sustained, regulated rate. The free fatty acids enter the bloodstream and are taken up by tissues that can oxidize them directly for energy. Muscle, heart, liver, tissues with abundant mitochondria run preferentially on fat oxidation during hibernation. Beta oxidation, the metabolic pathway that strips 2 carbon units from fatty acid chains and feeds them into the citric acid cycle, runs quietly and continuously through the winter months. The liver goes further. It converts some of the free fatty acids into ketone bodies, acetoacetate, beta-hydroxybutyrate, acetone, which are exported into circulation and used as fuel by tissues that can accept them, including the brain, which under normal conditions relies almost entirely on glucose. A cell receiving beta-hydroxybutyrate in the blood takes it up through a transporter, converts it back to acetyl-CoA inside the mitochondrion, and feeds it directly into the citric acid cycle. The process is clean and efficient. No nitrogen produced, no muscle protein consumed. The fuel comes from fat, runs through the mitochondria, and leaves as carbon dioxide and water. The muscle fiber is not touched. This is the qualitative distinction. It isn't that the hibernating animal is starving less severely, it's that it's running a different metabolic program, one organized around fat as primary substrate, with ketones as the secondary currency, and muscle protein as something the chemistry is specifically arranged to avoid. Human fasting moves in this direction too. After several days without food, ketone production increases in humans, muscle proteolysis slows somewhat, and the body's reliance on gluconeogenesis from amino acids decreases as ketones begin supplying the brain's energy needs. Some of what hibernators do, human metabolism can approximate. But approximation is not the same as the genuine article. The depth of fat reliance, the suppression of muscle catabolism, the sustained months-long maintenance of this metabolic state without the degradation of lean mass that extended human fasting produces. These don't fully cross over. What allows the hibernator to hold the fat fuel program so consistently without drifting back toward muscle consumption involves hormonal regulation, specifically the sustained suppression of the signals that would otherwise trigger significant muscle proteolysis, and the maintained sensitivity of fat tissue to lipolytic signals, even at low body temperature. Adipose tissue in a hibernating ground squirrel continues responding to hormonal signals for lipolysis at temperatures that would blunt the same response in human fat cells. The tissue has been prepared, again in that pre-hibernation window, to remain functional in cold. It releases fatty acids on schedule, the fuel keeps flowing, and the muscle, downstream of all this, receives the signal that it is not needed as a substrate. Not today, not this month. The hunger is real. The energy deficit is real. The extraordinary thing is simply that the body has arranged itself so that neither of those facts requires consuming what it spent the summer building. Conservation, not deprivation. The cell knows the difference. 7 days. That's roughly how long it takes for measurable muscle loss to appear in an otherwise healthy young adult placed on complete bed rest. Not a sick person, not someone elderly or nutritionally compromised. A healthy person, well-fed, simply horizontal and not moving. By day 7, the cross-sectional area of lower limb muscles has decreased detectably in imaging studies. Protein synthesis rates have dropped. The ubiquitin proteasome system has upregulated. The biology we've spent several chapters tracing in the context of disuse, the flagging of contractile proteins, the quieting of mTOR, the tilt toward net breakdown, is running in a healthy body after 1 week of not standing up. 7 days is not a long time. It is shorter than most hospital admissions for serious illness. Shorter than a typical recovery from major surgery. Far shorter than an ICU stay for a patient on mechanical ventilation. The timeline of medical immobility and the timeline of disuse atrophy are not parallel. They overlap almost completely from the very first days in ways that the clinical system has only recently begun to treat as urgently as it treats the primary illness. The data from Bedrest studies, accumulated over decades now, tells a consistent story. 1 week, measurable atrophy begins, force production decreases, insulin sensitivity drops. 3 weeks, lower limb muscle mass reduced by roughly 10% in healthy subjects. 6 weeks, the atrophy in some studies approaches what would be expected from 10 to 15 years of normal aging. The rate is not linear. The losses are steepest early when the body is responding to the sudden absence of loading signals and slow somewhat as a new reduced equilibrium is reached. But slow is not stop. The deficit accumulates. The physical sensation of this, the hollow lightness in limbs that have been still too long, the effort required for what used to be effortless, is something any person, who has spent significant time in a hospital bed, will recognize without needing it described. What they may not know is that by the time they noticed it, the biology had been running for days before the sensation became legible. ICU, acquired weakness, is a clinical entity now, recognized and named. Patients who survive critical illness, sepsis, acute respiratory distress syndrome, multi-organ dysfunction, prolonged mechanical ventilation, emerge from the ICU with a specific pattern of muscle loss and neuromuscular dysfunction that goes beyond what the underlying illness would explain. The immobility of critical care contributes independently. Sedation, which reduces motor activity nearly to zero. Neuromuscular blocking agents used to facilitate ventilation. The systemic inflammatory response, which has direct catabolic effects on muscle protein through inflammatory cytokines like TNF-alpha and interleukin-6. Corticosteroids used to manage inflammation, which suppress muscle protein synthesis. The combined insult produces atrophy at rates that exceed ordinary disuse atrophy, sometimes dramatically. Survivors of prolonged critical illness sometimes describe the body they return to as a different body, not in a metaphorical sense, functionally, measurably different. Some never fully recover the muscle mass they had before admission. Some return to a level of physical function that, 5 years later, is still below their pre-illness baseline. There are patients for whom surviving the ICU is not the end of a medical crisis, but the beginning of a years-long process of physical reconstruction, incomplete, requiring rehabilitation resources that are unevenly available and unevenly prescribed. The medicine has gotten very good at keeping people alive. What happens to their muscle while it does that has received, until recently, considerably less attention? This is changing. The field of ICU rehabilitation, early mobilization, passive and active exercise initiated within the first days of critical care admission when clinically feasible, has accumulated evidence over the last 15 years suggesting that moving patients earlier, even minimally, attenuates some of the muscle loss. Getting a ventilated patient to sit at the edge of the bed, passive range of motion exercises performed by physiotherapists. Neuromuscular electrical stimulation, electrodes placed on the skin over large muscle groups, delivering low-level electrical current that produces involuntary muscle contractions, bypassing the need for voluntary motor activity. The electrical stimulation approach is interesting for what it reveals about the mechanism. The muscle doesn't need the patient to be awake or cooperative or even conscious. It needs the mechanical signal, the contraction, the loading, the activation of the downstream pathways that tell the cell a muscle is being used. Neuromuscular electrical stimulation provides a version of that signal artificially, and studies show it reduces atrophy in sedated ICU patients compared to no stimulation. Not eliminates, reduces. The intervention is real and the effect is real, and both are modest. Protein supplementation helps too, to a degree. Ensuring adequate amino acid availability, particularly leucine, which has a specific mTOR activating effect independent of growth factors, supports the synthetic side of the equation. But protein availability alone, in the absence of mechanical loading, cannot overcome the suppression of synthesis that disuse produces. The substrate is there, the signal to use it is not. What medicine is working against in the immobilized patient is a biology that was not designed for the context it's operating in. The cellular response to disuse, the upregulation of muscle protein breakdown, the downregulation of synthesis, the ancient arithmetic that says don't carry what you don't use, evolved in an environment where prolonged immobility meant something was badly wrong, and in that environment, conserving energy by dismantling unused muscle was adaptive. The biology was right for the world it was written in. It is less right in an ICU in the 21st century, where the immobility is enforced and temporary, and the patient intends to use their muscles again if they survive. The cells don't know the intention. There is a particular frustration, quiet, the kind that accumulates over reading rather than arriving suddenly, in the gap between what medicine can now do for acute illness and what it can currently do for the muscle loss that accompanies treatment. The monitoring is thorough, the pharmacology is sophisticated, the surgical technique in many specialties is extraordinary, and the muscle continues to thin day by day because the fundamental problem, how to preserve muscle tissue in a body that isn't moving, remains only partially solved. The signals that would need to be sent to hold the muscle the way it is held in another kind of body during another kind of stillness are not yet something medicine can fully replicate. The electrical stimulation approximates one of them. The protein supplementation approximates another. The early mobilization tries to restore the real thing as soon as possible. None of it adds up to what that other biology does so quietly, without effort, without intervention, without any awareness that it's doing anything at all. The gap is not a failure of attention or care. It is a measure of how much the hibernating body knows that we don't yet. Inside a muscle fiber, there are lipid droplets. Not in every fiber, not always, but in hibernating mammals, ground squirrels, bears, certain bats, the muscle cells themselves accumulate small fat deposits during torpor, not in the adipose tissue surrounding the muscle. Inside the fiber, the fat is stored at the site where it will be burned, a few nanometers from the mitochondria that will oxidize it, available without transit through the bloodstream, without waiting for lipolysis in distant adipose depots to release free fatty acids into circulation. Intramuscular lipid. The term is precise and slightly ungainly, which is somehow appropriate for a thing that sits in an unexpected location, doing an unexpected job. In humans, the same phenomenon has a different story. Elevated intramuscular lipid in human muscle is associated with insulin resistance. It appears in obesity, in type 2 diabetes, in metabolic syndrome. The lipid droplets interfere with insulin signaling pathways inside the fiber through mechanisms involving diacylglycerol and ceramide intermediates that activate kinases which phosphorylate insulin receptor substrates in ways that reduce their function. The fat inside the human muscle cell is, in this context, a marker of something going wrong. It appears in imaging studies and biopsies as a sign that the cell's metabolic machinery is under a kind of chronic stress it wasn't designed to handle. Same molecule, same location. Opposite meaning. The hibernating muscle cell is not insulin resistant. Its intramuscular lipid is not a dysfunction. It is provisioning. The fat is there because the cell will need it, stored close because proximity reduces the energy cost of retrieval, cleared efficiently during arousal bouts when mitochondrial activity spikes and the stored lipid is oxidized to run the cell through its brief return to high activity states. The cell knows how to use it. What determines whether intramuscular lipid is provision or pathology isn't the molecule itself, it's the metabolic context surrounding it. The enzyme activity, the mitochondrial density and function, the hormonal milieu, the rate at which the fat is being turned over versus accumulating. A muscle cell with high mitochondrial capacity and active beta oxidation can manage intramuscular lipid efficiently. A muscle cell with reduced mitochondrial function operating in a context of chronic caloric excess and low activity cannot. The hibernator's muscle has been prepared for the fat it's carrying. The infrastructure for using it is in place before the fat arrives. I find myself sitting with that word, prepared, longer than the sentence warrants. There's something in the biology of pre-hibernation preparation that keeps surfacing in different forms. The membrane lipids changed in advance of the cold, the fat reserves built before the fast, the intramuscular lipid accumulated before it's needed. The body making decisions months before the consequences arrive. The same molecule in two different bodies, in two different metabolic contexts, doing two entirely different things. Biology doesn't deal in inherent meanings, only in circumstances. There is a dial and it turns slowly, not a metaphor for anything dramatic, just the image that keeps returning when I read about what thyroid hormones do during hibernation. A dial being turned by careful degrees over days and weeks until the number it points to is so low that every tissue in the body has had time to adjust to what the new number means. Nothing switches off, nothing crashes. The system recalibrates tissue by tissue, receptor by receptor, gene by gene, until the body is running at a fraction of its former rate, and the fraction feels, from the inside of the biology, entirely organized. The hypothalamic pituitary thyroid axis governs this. The hypothalamus releases thyrotropin-releasing hormone, which travels through the portal blood supply to the anterior pituitary, which releases thyroid-stimulating hormone into general circulation, which reaches the thyroid gland and stimulates the production and release of thyroid hormones, primarily thyroxine, called T4, and in smaller amounts, the more biologically active triiodothyronine, called T3. In peripheral tissues, T4 is converted to T3 by enzymes called deiodinases, and it is T3 that enters cells and produces most of the downstream effects associated with thyroid hormone action. In hibernating mammals, measured levels of T3 and T4 fall significantly during torpor. TSH falls. The entire axis turns down together, not through failure but through coordinated suppression, the hypothalamus reducing its TRH signal, the pituitary responding accordingly, the thyroid following. The reduction is not subtle. In some hibernating species, circulating thyroid hormone levels during deep torpor are a fraction of what they were in late summer. The dial has moved considerably. What thyroid hormones do in normal concentrations is, broadly, set the metabolic rate of every tissue that can respond to them, which is most tissues. T3 enters cells, crosses the nuclear membrane, and binds to thyroid hormone receptors that are already sitting on DNA, waiting. These receptors are transcription factors, proteins that, when bound to their Leydig end, change the rate at which specific genes are transcribed. The genes affected by thyroid hormone action include those coding for enzymes in oxidative phosphorylation, for proteins involved in calcium handling in muscle, for the sodium-potassium ATPase pumps that consume a substantial fraction of a cell's resting energy maintaining ion gradients across membranes. When T3 is present at normal levels, these genes are expressed at rates that support a high metabolic throughput. The mitochondria run fast, the pumps run continuously. The cell burns fuel at a rate consistent with an active mammal maintaining body temperature in a variable environment. When T3 falls, the expression of these genes changes. The mitochondria run slower, the pumps reduce their activity. The basal metabolic rate, the energy the cell consumes simply to exist, decreases. This is not the cell breaking down. This is the cell reading a different set of instructions. The nuclear action of thyroid hormone is, I think, one of the more quietly remarkable things in endocrinology. The idea that a small iodinated molecule produced in the neck travels through the bloodstream, enters cells throughout the entire body, crosses into the nucleus and physically changes which genes are being read. The hormone doesn't carry the instructions. The instructions are already in the DNA. The hormone changes which passages are open, and when the hormone falls, different passages open. For muscle, the downstream consequences of thyroid suppression during hibernation include a reduction in the basal rate of protein turnover. This connection between metabolic rate and protein turnover is not incidental. Protein synthesis is energetically expensive, consuming a significant fraction of a cell's total ATP budget. When the cell is running at reduced metabolic rate, when the mitochondria are producing ATP more slowly and the overall energy economy is contracted, protein synthesis is among the processes that scale down accordingly. Protein degradation scales down too, for related reasons. The proteasome requires ATP to function, and at reduced metabolic rates, the ATP available to run it is reduced. The entire turnover cycle slows. Both sides of the balance contract together at a rate set partly by how much thyroid hormone is telling the cell's mitochondria to produce. This is the coordinated part, the part that makes hibernation metabolically coherent rather than simply diminished. The thyroid axis doesn't just reduce energy expenditure in isolation. It recalibrates the rate at which every energy-consuming process runs, so that the whole system arrives at a new, lower operating point that is internally consistent. The muscle doesn't lose protein faster than the reduced synthetic rate can compensate, because both rates have been turned down by the same upstream signal. The dial moves everything together. What the cold does to cells, the membrane changes prepared for. What the fast does to energy supply, the fat metabolism answered. What the inactivity would do to protein balance, the thyroid suppression partially counteracts by reducing the rate at which both synthesis and degradation run, so the tilt between them remains manageable. None of this is the whole answer. The thyroid suppression interacts with AMPK signaling, with the nitrogen recycling, with the membrane fluidity changes, with the arousal cycle synthesis windows. The biology is not a single mechanism, but an arrangement of mechanisms, each one contributing to a preservation that none of them could achieve alone. But the thyroid quiet is foundational. Without the metabolic slowdown it enables, without every tissue in the body reading the lower number on the dial and adjusting accordingly, the other mechanisms would be working against a resting energy expenditure that the fat stores couldn't sustain for months. The dial comes down, the tissues listen, the body enters a register so low, so internally consistent, so far from what it was doing in October that describing it as the same animal seems almost imprecise. It is the same animal, running very slowly on purpose. You are somewhere between 3 and 4 hours into sleep right now, if the timing is right, maybe less. Maybe you're reading this at an edge, half in and half out, the words arriving more slowly than they did an hour ago. Either way, wherever you are in the night, there is a version of this that applies to you, not as metaphor, as current fact. Your body at 3 a.m. is not resting. The word rest implies suspension, a pause, a waiting. What's actually happening in the hours between midnight and dawn is something more like a parallel shift, a different kind of work beginning as the day's work ends. The organs that were running support operations through your waking hours are now, in some cases, running at their highest activity of the full 24-hour cycle. The brain that managed your conversations and your decisions and your anxieties is doing something else entirely with the quiet you've given it. The muscle that moved you through the day is being tended to in ways it cannot be tended to while it's being used. The liver is one place to begin because most people don't think of the liver as a nocturnal organ, and in a meaningful sense it is. Hepatic metabolic activity follows a circadian pattern. The liver has its own internal clock, synchronized to the master clock in the suprachiasmatic nucleus of the hypothalamus through hormonal and neural signals, and its peak activity for certain processes coincides with the sleeping hours. Glycogen synthesis, the storage of glucose as glycogen, building the reserve that will supply blood sugar through the next morning's fast before breakfast, runs robustly during sleep. Cholesterol synthesis follows a circadian rhythm that peaks at night, which is why statins, drugs that inhibit cholesterol synthesis, are often prescribed for evening use. The liver's detoxification processes, cytochrome, P450 enzymes, metabolizing drugs, processing metabolic byproducts, clearing compounds that accumulated during waking activity, run with circadian variation, some peaking nocturnally. The liver at 3 a.m. is busy, not frantically, not with any urgency you could detect from outside, quietly, chemically busy, doing the accounting and restocking and clearing that a full day of metabolic activity has left undone. Your weight in the bed hasn't changed. Your breathing is slow. There is no outward sign of any of this. In the muscle, in the fibers that carried you through yesterday, that absorbed whatever stress or movement or stillness the day asked of them, the repair processes we've traced across the last several chapters are running. The anabolic window that slow-wave sleep opens is either fully open right now or beginning to narrow, depending on where you are in the night's architecture. Growth hormone has pulsed. IGF-1 is circulating. The mTOR pathway in muscle cells is running at a higher rate than it will during any waking hour. Ribosomes that slowed through the afternoon are translating protein again, myosin, actin, the cytoskeletal proteins that give fibers their structure, the metabolic enzymes that allow the fiber to generate energy efficiently. The small injuries of the day are being addressed. Microdamage to contractile proteins, the ordinary wear of a body that moved, that lifted, that held tension through hours of sitting in ways the spine didn't love, is being cleared by the same autophagic processes that run in the hibernating squirrel, scaled to a different metabolic context but following the same cellular logic. The cell is doing housekeeping. The proteasome is running at its nighttime rate. The balance between synthesis and degradation is, right now, tilted toward building. This is the ordinary miracle, not a dramatic one, not one that announces itself, just the biology doing what it was always doing, in the dark, while the mind that inhabits the body is somewhere else entirely. The brain, meanwhile, is being washed. The glymphatic system, discovered and characterized only in the last 15 years or so, which is striking given how long we've been studying the brain, is a network of channels that surrounds the blood vessels of the central nervous system, through which cerebrospinal fluid flows and clears the metabolic waste products that accumulate during waking neural activity. Beta amyloid, tau protein, the biochemical byproducts of a day of thinking and perceiving and feeling. During sleep, and specifically during slow-wave sleep, the space between brain cells expands, by some estimates by 60% compared to waking, and glymphatic flow increases dramatically. The brain, during sleep, is cleaning itself in a way it cannot clean itself while it is awake and busy and taking up the space that the fluid needs. I find it hard to hold this fact at a distance. The idea that sleep is, among many other things, the only time the brain has to clear its own waste, that the metabolic debris of consciousness is managed not during consciousness but in its absence, is the kind of biological fact that changes the texture of lying down. The darkness isn't absence, it's maintenance. You're breathing right now. If you're still reading, if the words are still arriving, is slower than it was an hour ago. The respiratory rate drops during deep sleep. The tidal volume, the amount of air moved with each breath, changes. The body is using less oxygen than it was, not because it has less to do, but because the doing it's occupied with requires less oxygen than waking activity. The metabolic rate during deep sleep is genuinely lower than during quiet waking rest. The dial has turned, in your body too, in your own small way, not as far as the hibernator's dial, not nearly, but the same direction. Somewhere in the adrenal cortex, two small glands sitting atop the kidneys, synthesizing steroid hormones from cholesterol, cortisol production is beginning to increase. Not rapidly, not urgently, a slow rise, following the circadian rhythm that has been timed over your life and over evolutionary time to anticipate the return to consciousness. Cortisol will be near its daily peak by the time you wake. It will raise blood glucose by stimulating gluconeogenesis in the liver. It will increase alertness through mechanisms involving the noradrenergic system. It will prepare the body for upright posture for the metabolic demands of a day. It will also, at the concentrations that waking cortisol reaches, begin to shift muscle metabolism back toward the catabolic direction. The anabolic window will close. The repair work of the night will hand off to the activity of the day. This is not a failure of the system. It is the system. The repair requires the quiet. The quiet requires the day to end. The cortisol rise is what makes the day possible again. The biology is not trying to undermine what it built during the night. It is transitioning in an orderly way between two different operating modes that a human body needs in sequence daily to function. The window closes so that you can open your eyes. Right now, it is still open. The liver is running. The glymphatic channels are flowing. The ribosomes in your muscle cells are translating. The growth hormone has done what it came to do. The brain is somewhere in its cleaning cycle, somewhere in the slow waves or the dreaming or the transitional states between, doing work that has no waking equivalent. You are not resting. You are being repaired. And the repaired version of you, the one that will exist by morning, slightly more intact than the one that lay down, is being assembled right now, in the quiet, without your help, without your awareness, without any requirement that you understand or appreciate or even know about any of the processes running in the dark of your own body. They have been running every night of your life, long before you knew what they were. They are running now. Something changes before the animal wakes, not a decision, not a conscious process of any kind. A shift in the chemistry, small at first, then gathering, that begins while the animal is still deeply torpid, still at a body temperature that would render a human unconscious, still technically unreachable by the ordinary criteria of wakefulness. The arousal begins in the brain, specifically in the hypothalamus, where the master circadian clock and the thermoregulatory centers share real estate, where the signals that have been suppressing normal waking physiology for months begin, for reasons that are not entirely understood, to change. Thyroid-stimulating hormone begins to rise. The thyroid axis, which we watched turn down so carefully weeks or months ago, begins its slow return. T3 levels start to climb. The dial, which we left at its lowest setting, begins moving back. Core temperature is still low, 3 degrees Celsius. 4. The metabolic rate is still a fraction of summer levels. But something has been decided, in the only sense that biology decides things. A threshold crossed, a feedback loop tipped, a cascade begun that will not reverse. Thermogenesis starts in brown adipose tissue. Brown fat, different from the white adipose tissue that stores energy, is specialized for heat production. It contains mitochondria in unusual density, and those mitochondria express a protein called uncoupling protein 1, which allows the proton gradient built by the electron transport chain to dissipate as heat, rather than drive ATP synthesis. The mitochondria run without producing their usual currency. They produce warmth instead. This is the first heat. Internal, generated before any muscle has contracted, before any shivering has begun, rising from the fat deposits around the heart and between the shoulder blades. Then the shivering starts. Shivering is skeletal muscle contracting rapidly, involuntarily, out of phase. The fibers of opposing muscle groups firing in rapid alternation, generating heat through the inefficiency of the contractions rather than through any useful movement. The animal is not trying to move. The muscle is being used as a furnace. The ATP consumed in those contractions is converted almost entirely to heat, and the heat travels outward through the tissue, raising core temperature by degrees, one shiver cycle at a time. The muscle that has been still for months, preserved through the nitrogen recycling and the metabolic suppression and the membrane changes and the careful management of protein turnover we've spent this whole video tracing, is firing now. Before the animal is conscious, before the eyes open, before there is anything that could be called awareness of the cold or the den or the approaching spring. The muscle acts before the mind returns. I find that sequence difficult to move past quickly. The body re-engaging its own machinery before the self that inhabits the body is present to experience it. The muscle firing in service of warmth while the cortex is still, by any practical measure, offline. Life insisting on its own return through a biological grammar that doesn't wait for the animal to be ready. There is nothing surprising about this from a mechanistic standpoint. Shivering thermogenesis is a brainstem reflex driven by temperature-sensitive neurons that don't require cortical input. The muscle doesn't need the bear to be awake, it just needs the signal. But knowing the mechanism doesn't fully dissolve the strangeness. As temperature rises, the hormonal landscape shifts further. Growth hormone surges during arousal, a large pulse, measurable, the pituitary gland releasing what it has been holding back. Cortisol rises, the adrenal axis wakes ahead of the animal. Thyroid hormones continue climbing. The metabolic rate begins its return toward the summer levels, the mitochondria recoupling, the sodium-potassium pumps resuming their full activity, the cells returning to their higher energy operating register. Muscle protein synthesis during arousal and in the hours immediately following runs at elevated rates. The synthetic window that each torpor arousal cycle has been providing through the winter, the brief returns to metabolic normalcy that the chapter on slow proteolysis described, has now extended into full waking. The ribosome runs at summer speed. The repair that was managed in fragments through the winter can now proceed continuously. The body takes stock of itself. The deficit, if there was one, begins to close. By the time full consciousness returns, by the time the animal is neurologically present in any sense we'd recognize, the muscle has already been working for some time. The warmth has already been generated. The temperature has already climbed. The body arrived before the self did. This is how hibernation ends. Not with a dramatic waking, not with a gasp or a sudden awareness. A slow chemical return, organ by organ, tissue by tissue, following a sequence that has been followed in this species for longer than the species has had its current name. The grammar of emergence, precise, ordered, old. There is a question underneath everything this video has been moving toward, and it isn't a dramatic one. It doesn't resolve into a clean answer. It's more like a shape that keeps appearing in different biological contexts. In the bear's den, in the squirrel's burrow, in the specific hormonal architecture of your own slow-wave sleep. And the shape is this. What if stillness, for certain kinds of bodies, under certain kinds of conditions, is not the absence of protection, but the form protection takes? Not rest as waiting, rest as repair, not dormancy as failure to act. Dormancy as the most sophisticated biological action available. The hibernating mammals we've been tracing through these chapters, the brown bear with its recycled nitrogen, the ground squirrel with its tilted proteolysis and its weekly arousal windows, the membranes rebuilt, lipid by lipid, before the cold arrived, are not simply surviving winter through inactivity. They are running an alternative biological program, one that achieves in months of stillness what a waking body achieves in months of activity through entirely different means. The muscle is preserved not despite the stillness, but through mechanisms that the stillness enables. The conservation is active, the quiet is doing something. And then there is the question of what your sleep is, not rhetorically, actually, what it is biologically in the context of the evolutionary history of dormancy in mammals. Whether what happens in your body between roughly 11 p.m. and 6 a.m. is a compressed, warmer, shorter version of something that runs in longer and colder forms in other species. Whether the growth hormone pulse and the glymphatic flush and the mTOR, activation in sleeping muscle, and the reduced protein degradation of the nighttime hours represent a genuine biological continuity with the deeper dormancy of hibernating species. A family resemblance, written in the same hormonal language, running the same cellular logic at a different magnitude? The honest answer is that we don't fully know. What we do know is that the molecular mechanisms overlap considerably. The same pathways, the same hormones, the same cellular receptors doing the same things in response to the same signals. The suppression of cortisol during deep sleep, the rise of growth hormone, the shift toward anabolic muscle metabolism. These are not analogous to what hibernating animals do. They are, in many cases, the same thing, running at reduced amplitude and reduced duration in a body that wakes every morning rather than every spring. Whether this makes human sleep a vestigial form of something older, a hibernation compressed by evolutionary pressures into a nightly cycle rather than an annual one, stripped of its most extreme features but retaining its core biological logic, is a question comparative biologists approach carefully. The evidence is suggestive rather than definitive. The shared mechanisms are real. The evolutionary continuity between mammalian sleep and mammalian torpor is a genuine area of active research, not a speculative idea. But the claim that human sleep is hibernation light would be an overstatement, and I find I don't want to overstate it. What I want to stay with instead is the continuity at the cellular level. The fact that when you sleep deeply, sufficiently, in a body that has been given the darkness and the time it needs, your muscle tissue enters a state of relative preservation. The proteolysis slows slightly. The synthetic rate rises. The nighttime hormonal environment is specifically organized around rebuilding what the day dismantled, not perfectly, not with the extraordinary efficiency of a hibernating bear that emerges from 7 months without food with its muscle largely intact, but genuinely, measurably, in a direction that matters for every tissue in your body. The body that wakes after a full, deep night of sleep is different from the one that lay down, slightly more repaired, slightly more intact. The difference is not always perceptible. You don't feel your muscle protein turnover shifting. You don't sense the glymphatic channels flushing the day's metabolic waste. But the difference is real, and it accumulates. Night after night, the repair runs. Night after night, the window opens and something is done with it. And the body that wakes after nights of insufficient or fragmented sleep, the one that didn't spend enough time in slow wave sleep, that had its growth hormone pulse diminished by alcohol or light or anxiety, or the particular modern difficulty of lying down while the mind keeps running, that body is also different, slightly less repaired. The window opened and closed before the work was finished. The deficit is small on any given night and significant across months and years. This is not a judgment. It's a biological description of a gap between what sleep can do and what it is sometimes allowed to do in the context of lives that don't always make space for it. The hibernating bear makes no such choice, but then it also has no choice. The biology runs because the season requires it, and the season allows no negotiation. There is a kind of, not envy, but noticing, in recognizing how complete the protection is when the animal has no option but to submit to the stillness entirely. No disruptions, no abbreviated arousals, no checking anything. Just the den, and the months, and the chemistry running undisturbed until it has done everything it needs to do. Your sleep is not that. It cannot be that. But the biology underneath it is the same biology. The hormones are the same hormones. The cellular receptors are the same cellular receptors. The logic, that stillness handled correctly with the right molecular preparation and the right hormonal environment and the right duration, is protective rather than merely passive, is as true for your sleeping body as it is for the torpid ground squirrel, 3 metabolic states below ordinary wakefulness. Your body knows how to do this. It has been doing it every night. Right now, in whatever dark you're in. The muscles that move today are being tended to. The liver is running its nighttime chemistry. The brain is in its cleaning cycle or approaching it. The hormones of repair are either already circulating or preparing to. The window is open or opening, and the biology that has been running in various forms for hundreds of millions of years of vertebrate evolution is running in you, in this ordinary darkness, doing what it was always designed to do. The cortisol will rise eventually, the morning will come. But that's not now. Right now the stillness is working. The muscle is held, the repair is running. The body, in its stillness, is keeping you.